This is Daniel Millard, and welcome to our webinar describing the design of evoked assays for evaluating functional neurotoxicology with multiwell microelectrode array technology. In vitro models of rodent and iPSC-derived neurons have been used extensively to assess functional neurotoxicology and safety pharmacology, whereby changes in the functional activity of neural circuits inform the risk of a particular chemical or drug. In this assay, the cells are seeded into a multiwell MEA plate and allowed to attach to the surface and form a functional network. Upon maturation, a label-free measurement is acquired with the Maestro platform, providing endpoints on the activity, synchrony, and oscillations of the neural network in each well. In this example raster plot, which shows the spike times for each electrode in a well across time, we can note the significant change in activity following the addition of picrotoxin, a known GABA antagonist. The oscillations become slower and more pronounced, while the overall activity remains similar. In participation with the recent Hesse-Neutox study aimed at detecting seizure-genic risk, spontaneous endpoints of neural function were used to evaluate a panel of 12 compounds across multiple cell types. While many compounds were detected correctly, some seizure-genic compounds were missed with the spontaneous endpoints. For example, chlorpromazine and amoxapine showed similar trends to the anti-epileptic drug phenytoin, and linopyridine and strychnine exhibited biphasic trends with only modest responses at intermediate concentrations. Today we will explore if stimulation-based assays can provide additional information for the assessment of neurotoxicology and safety pharmacology, specifically seizure-genic risk. A spontaneous assay quantifies changes in the neural activity, which may differ across wells or cell models. An evoked assay uses a controlled stimulation input to produce a controlled output and can offer many benefits, namely enhanced reliability across wells or an accelerated assay timescale. Stimulation also allows new evoked assay endpoints, or patterns of stimulation can be designed to specifically target a known biological function. We'll briefly touch on each of these three aspects today. First, we will highlight the differences between the two modes of stimulation, namely electrical stimulation via the embedded microelectrodes in each well, and optical stimulation that engages light-sensitive ion channels that can be genetically expressed within the cell population. An electrical stimulation event activates the neurons around the stimulated electrode, and the activity then propagates throughout the cellular network. Dependent on the mode of delivery, expression of the light-sensitive ion channel, or opsin, here denoted by the purple cell boundaries, can occur across the entire population of neurons, or a genetically defined subset linked to a promoter sequence included in the optogenetic construct. Here, a flash of light activates the expressing neurons, and the activity propagates throughout the network. A wide variety of opsins are available within the scientific community, and each can be controlled through the Lumos multiwell optical stimulator. Opsins, such as channel rhodopsin, exist for depolarization or excitation of neurons, whereas ArchT, for example, can hyperpolarize a genetically defined subset of the cellular network. With 4 independently controlled LEDs per well in the 48-well Lumos, opsins with distinct spectral responses can be activated separately. In this case, Chronos is a green-shifted opsin relative to channel rhodopsin, and activation of crimson is red-shifted. Finally, each opsin has unique channel kinetics, allowing for rapid or sustained activation. Chronos has optimized channel kinetics to allow for high-frequency stimulation. The Lumos can provide high-speed optical stimulation at millisecond precision to take advantage of these unique opsin features. The first advantage of a stimulation-based assay is to standardize the network activity across wells and conditions, thereby reducing well-to-well variability. Furthermore, the frequency and magnitude of network events are often coupled. Here, optogenetic stimulation controls the network event frequency to isolate the network response magnitude. As mentioned previously, stimulation-based assays can also provide new evoked endpoints unavailable in a spontaneous assay. For a seizure-genic assay, we are interested in compounds that alter the balance of excitation and inhibition within the network. In vitro cortical models consist of interconnected excitatory and inhibitory neurons that work together to create network events. A network event begins with excitation and ends with inhibition. The timing and intensity of the subsequent event can provide important information on the excitatory and inhibitory balance. We can use an evoked assay with paired stimulation to probe this network property. In this paired pulse assay, we deliver pairs of stimulation separated by a brief delay and compare the response of the two stimuli. As we vary the inner stimulus interval, we note that a short delay leads to a smaller response to the second stimulus. This is quantified as a paired pulse ratio of less than 1. With a long delay, the inhibition has subsided and the response to the second stimulus is the same as the first, such that the paired pulse response ratio is 1. We can map the response ratio over a range of inner stimulus intervals for a vehicle control and a positive control, in this case picrotoxin. Whereas the vehicle control requires about 5 seconds for the inhibition to subside, the picrotoxin positive control returns to a response ratio of 1 with an inner stimulus interval of 1 second, indicating that picrotoxin shifted the balance towards excitation. We chose the 1 second inner stimulus interval as the assay point and screened the same compounds discussed in the Hesse-Neutox study earlier. Here, picrotoxin showed a dose-dependent trend towards overexcitability, whereas the negative control, acetaminophen, showed no change. Importantly, the vehicle and positive controls behaved consistently across plates, with the data from 2 plates shown here. Linopyridine and chlorpromazine are 2 of the seizure-genic compounds mentioned earlier that were neuroactive in the spontaneous assay, but did not show evidence of seizure-genicity. Here, however, both compounds showed a shift towards overexcitability at low concentrations. It's worth mentioning here that this assay utilized a single measurement read after dosing without the use of a baseline measurement. We'll end here with an exploratory example of a targeted assay, whereby the pattern of stimulation is designed to target a specific aspect of the biology. Carbamazepine, like many other anti-epileptic drugs, acts by stabilizing the inactive state of the voltage-gated sodium channel, such that it blocks high-frequency firing without affecting normal brain activity. Here, cortical neurons were transduced with Chronos, an opsin with extremely fast kinetics, allowing high-frequency stimulation mimicking the activity occurring in a seizure. When dosed with carbamazepine, there was a loss in evoked spike magnitude and precision as the high-frequency pulse train continued. This brief example highlights that optogenetic stimulation offers flexibility in designing precise, patterned inputs to probe neural function and perform mechanistic assays. In summary, evoked assays with electrical or optogenetic stimulation can increase assay reliability, accelerate assay timescale by elevating activity levels, decouple endpoints related to network activity, such as the network burst frequency and duration, produce new evoked endpoints of neural circuit function, stimulate advanced patterns of activity, such as high-frequency seizure-like responses. Further, the combination of optogenetics and the Lumos enable well-wide illumination for simplified assay execution, excitation or suppression of genetically targeted subpopulations, and neural control on a wide range of temporal scales. Thank you.
Thank you for watching this webinar with Dr. Daniel Millard. If you have any questions for Dr. Millard, please email them to questions@axionbio.com.